The overall goal of this procedure is to prepare single living photoreceptor cells for fluorescence imaging. This is accomplished by first isolating a dark-adapted retina. The second step of the procedure is to isolate single living photoreceptor cells. The third step of the procedure is the transfer of the cells in an experimental chamber. The fourth step of the procedure is the placement of the chamber on the microscope stage. The final step of the procedure is the selection of a healthy cell for fluorescence imaging. Ultimately, results can be obtained that show real-time changes in cell metabolism, signaling, intracellular trafficking, or oxidation through imaging of the cell fluorescence.
This method can help answer key questions in the vertebrate photoreceptor field, such as clearance of toxic byproducts of the detection of light.
The implications of this technique extend toward the therapy of the diseases of the retina because can be used to study the photoreceptor cells from mouse models of the diseases.
Before beginning the protocol, you need to prepare various materials in advance. First, 35mm Falcon Petri dishes with just enough CellGuard elastomer to coat the dish need to be prepared a few days in advance so the elastomer can harden. The experimental chambers are modified 35mm Petri dishes with a 12mm diameter glass bottom. The experimental chambers need to be sticky so cells are immobilized. Add 200 microliters of 0.01% poly-L-lysine or poly-L-ornithine to the bottom of the chamber.